Breathe.
Alright, so we're going to be doing the dry step. Here is our rack of columns and tubes. So this is the same thing, we've just walked over to a centrifuge that has enough space to handle this all. So we have 8 plus 6, so that's 14. So we're just going to do 7 on each side. So we always need to balance this thing. So 1, 2, 3, 4, 5, 6, and 7. And then we're just going to go to the other half. So this is 24. So we're going to go to 13. So 1 through 12, and then 13 through 24 for the next 7. So 13, 14, 15, oops, what's going in there, 16, 17, 18, and 19. Then we're just going to spin this for 90 seconds, 2 minutes. This is actually a really fast centrifuge, so we can get away with 90 seconds. Oh, just to make sure, I capped this. Always make sure this gets capped. So 1 minute, I guess we'll just do 2 minutes. Hit start, and this thing will gain speed. And then while this thing is spinning down, let's just do one more review of the whole mini prep process. I believe in repetition. So we start off with our overnight culture in our 24 well block. We spin that down in the large centrifuge. We get a pellet. We toss the supernatant into some bleach water. We tap it down on some paper towel in order to get rid of the excess moisture that's inside of there. We then take our P1 out of the refrigerator, put 250 microliters of P1 into each well. We covered it up with a plate seal, one of those clear plastic things, and vortexed it until our pellet resuspended. We wanted to check our pellet from the bottom and from the top to make sure that there's no chunks of bacteria sitting there. After we are satisfied that we've resuspended our pellet in the P1, we're going to add 250 microliters of P2. That's going to break open the cells. It's going to dissolve them. That means it's going to spill their guts, and the proteins and the cell walls are going to crack open, and it's going to clear the culture. And then after that, we have to rock that very gently when we add the P1. After that, we're adding 350 microliters of N3. That is going to be the neutralization buffer. That's what crashes everything out. That gets rid of the genomic DNA. That gets rid of the proteins, the cell wall, cell membrane, and it condenses or it precipitates it into some white gunk. And we need to make sure that we break that up real well. So you're going to start off with a very easy rocking, but as it gets more and more broken up, you can shake it more and more vigorously, so you get a bunch of little kind of like granules of precipitate. Otherwise, the interface between the N3 and the rest of the mini prep is going to stay intact, preventing further mixing, which will reduce the efficiency of your mini prep. After that, we spin it for 10 minutes to pellet all the junk that we don't want. We then pull up the supernatant, which has the plasmid DNA. We put it onto columns on the vacuum manifold. We then flowed that through. We then rinsed, or rather we washed with 500 microliters of PB. We then washed with 1,000 microliters of PE. We put our columns inside of these little tubes, and we spun for 2 minutes in order to dry them. That's where we're at right now, and now we are ready for the elution step. So all we got to do is pull out our tubes. And if we look, we can see there's maybe just a little bit of moisture at the bottom of these. You can barely even see it. But that's all that this drying step does. It gets rid of it. If we have any alcohol, residual alcohol in our mini preps, then the water can't really dissolve the DNA. So that's why we really want to make sure that the columns are completely dry. So pull out your tubes. And you want it to be double checking the order every time to make sure that you haven't mixed anything up. And then we're going to head over to my bench and start the elution. And we're going to elute in 50 microliters of water. This is just the typical elution volume. And after that, we'll have completed our mini prep. So we're going to open up all of our tubes. And these are our collection tubes. This is what we're going to elute into, just 1.5 mil epi tubes that we've already labeled. And we can put our columns into those tubes. And we reuse these, we don't throw those out, so we'll just hold on to them.
You're going to digest it a little bit, and then we're going to run the digester bill. I found that it's typically not worth running the first bill. Sure. If we turn out that there's nothing there,
so now all of our columns are inside of our tubes, and we're going to just open them all up so that we can get our water in. And we're going to be eluting with double deionized and autoclaved water. So we start off with, we buy double deionized water in a little box. You can also get it from the facility you work at probably. And then we autoclave that just to make extra sure that it was sterile. So this is the best water you could possibly get, at least for working in a lab. You'd never want to drink it because it's so pure that it would lyse all of the cells in your gut. It would throw off all the osmolarity. So, get our P200, set it to 50. If you're working with a really high copy plasmid, you can sometimes go up to 60, 75, 100 if you really want to. So then we have our water right here, and pull up 50, and we're going to try to aim as close to the center as we can, and we want to cover the entire membrane with water. And I'm trying to reuse the tip because it's a little bit faster that way, but if I did have to touch my tip to one of these columns, I would have to switch tips, just to make sure that I don't get any contamination. Because not only would I contaminate my mini preps, I'd be contaminating my water, and that's the kind of thing that would possibly get picked up in a PCR, and we use water in our PCRs, so you got to be extra careful about contamination here. A little bit of DNA goes a long way in a PCR. Okay, so eject the tip, cap the water, put it back. So the protocol says that you want to let your water sit inside of your tube for a little bit to start to dissolve the DNA before you spin it down. I don't actually give it time to wait because I figure that while I'm putting the water in, everything else is slowly dissolving, and by the time I actually get to the centrifuge and start my spin, it's already had plenty of time to dissolve. So if you're to look at the time tracker on the video, you'd see that by the time we even start spinning, it will probably already have been about 2 minutes. So now we got to put our tubes in here, and we got to be careful what we do with the tops. So this centrifuge has a little arrow pointing that way. That means this rotor is going to be spinning in the same direction as that arrow. That means we want the tops to be facing in the opposite direction. So always face your tops in the opposite direction of that arrow. So we're going to put 7 in, and I'm going to stagger mine a little bit. So I'm going to do 4 and then 3 just to give my tops enough room. So there's 4, then I'm going to skip 1, and then I'm going to do 3. And if I don't skip one, these tops slowly get more and more bent towards the center, which makes them more liable to snap off, which is no good. Especially in this centrifuge, it's a little bit of a higher powered centrifuge, so it snaps things off real easy. So 4 and 3. 1, 2, and 3. And then before I cap anything, I just give it a little spin and touch it with my finger to make sure everything is all nice and set. I cap it, and then I can just spin it for 30 seconds. Hit start. Wait for 30 seconds. So right now the water had plenty of time to dissolve the DNA that was on the column, and the spin step is going to force it through the column. The water is going to get collected in our epi tube, and from there we're going to be able to do mapping, we're going to QC it, we're going to sequence it, or we're going to go into an assembly, or we're going to do whatever we're going to do with our plasmid. So a couple more minutes, or I guess seconds. So as soon as the thing stops, the door lights up yellow. All centrifuges are different, and then you can open it. And then we're just going to take out our tubes. I actually take them out in the reverse order because it's a little bit easier. You can just grab the top. Look at the column, close your tube, double check that's right, make sure you have liquid, and then you can toss the column. So you're going to do that same process, do it slow a couple times. So top of the column is T5, top of the tube is T5. Do I have liquid? Yes. Top of the column. Look at the top of the column, T4, T4, liquid? Yes. And then we just do that for each time. So it's, it gets a little bit faster, but always double check. It's easy to get tube switches. You always want to catch a tube switch if you can. Everybody makes them, and they will really mess you up in the long run. So just 8 more samples, making sure I have liquid in all of these, and making sure that everything maps onto what it is supposed to map onto. So 6, 6 liquid, 5, 5 liquid, 4, 4 liquid, 3, 3 liquid, 2, 2 liquid, and last one, 1, 1 liquid. And now we have our whole array of tubes that have the plasmid DNA in it, and from here we go into QC. So that's going to be digesting it, we're going to be EcoBAM mapping it. After that, depending on what clones look good, we're going to send off for sequencing, and we're going to confirm the sequence of it. And you can do whatever else you want. You're going to go into assembly, you're going to do an EcoBAM transfer, you can PCR off of it, whatever. So that is how you do a 24-well block mini prep at the medium throughput. If you have, I would say, 6 or more, that's kind of my cutoff for doing it in a 24-well block. Otherwise, I'll just do it with 2. So let me know if you have any questions, and we'll see you soon.